Testing. All right. So let's get started. All right, so today we're talking about cells. Cytology is the study of cells. The root word cyto means cell. And cells are small enough that you will need a microscope to observe them, and they're measured in micrometers, which are 1/10,000th of a centimeter. Um, let's see. Some cells are large enough to be visible to the naked eye. For instance, the female egg cell is, um, if you were looking at a printed page, it would be about the size of a period on that page. So big enough to see, but still not really big enough for us to see. Neurons and muscle cells, while they're not uh, very wide, are very long. They actually run for in the case of some nerve cells, up to a couple feet in length. All right. Microscopy is using a microscope to view small scale structures. And for most tissues, we are going to need to stain them with dyes so that we can see them and get contrast between the different tissues and different cell types. So we can use a light microscope, and a light microscope produces a two-dimensional image by shining light through the specimen. It's your standard microscope. When you think of a microscope, that's the one you're thinking of. We also have electron microscopes where we bounce a beam of electrons off of the specimen, and we can get greater magnification and resolution than with a light microscope. Two common types are the transmission electron microscope that hits the subject with an electron beam through thin cut sections, and that'll get you a 2D image. And you can also use a scanning electron microscope that directs the beam across the surface of the specimen, and you can get 3D images with the scanning electron microscope. Those microscopes are incredibly expensive. All right. And so here on the left, you see the light microscope. Most of the images that we see are going to look like this. And you can see that they've used dyes or stains to give us contrast between the different types of tissue. In this particular tissue, you can see a lot of, looks like smooth muscle at the bottom, and then at the top is epithelia with cilia. In the middle is a scanning electron microscope, which gives us that three-dimensional image. This is zoomed in on the cilia. And then the transmission electron microscope gives us another two-dimensional image, again of the cilia, but we can get it in really high detail. So cells will vary greatly in their size and shape. For example, a red blood cell is between 7 and 8 micrometers, while the human egg cell has a diameter of 120 micrometers. Most are going to be microscopic, meaning you need a microscope to see them, and their shapes will vary. You can have spherical cells like the egg, you can have cube-like cells, column-like cells, you can have cylindrical cells like muscle. You can have disc-shaped cells or cells that have an irregular shape like sperm. All right, you can see the range of cell sizes here. Okay, I'm just going to move on. You can see the different types of shapes. Red blood cells have a biconcave disc shape. They look like a disc that's concave on two sides. Kind of looks like a raft, in my opinion. Nerve cells have an irregular shape as well. I mentioned sperm earlier, but they're both pretty irregular. Then we can have epithelia that are cube or column shaped. Connective tissue tends to be spherical, as is the human egg, and then muscle tends to be cylindrical or shaped like a cylinder. So the plasma membrane forms the outer barrier, the outer limit of the cell. It separates inside from outside. Okay, and There are modified extensions of the plasma membrane, such as cilia, which are those little hair-like attachments we saw a couple slides ago. There's a flagellum, which is that long tail on the sperm cell. Sperm cell is the only one that has a flagellum, and it's used to move the sperm cell. And then you can have microvilli, like on the epithelia in the small intestine. Microvilli increase the surface area because the entire point of the small intestine is to absorb nutrients, and so we want to have as much surface area as possible available to absorb nutrients. The nucleus is the largest structure in the cell, and it's surrounded by the nuclear envelope, which is it actually has two membranes surrounding it, and it contains the genetic information uh, in the form of DNA. The nucleoplasm is the fluid inside the nucleus. Yeah. So the cytoplasm is the fluid and all of the inclusions inside of a cell. So the cytosol is the fluid, and then the cytoplasm includes the fluid and everything suspended in it. Technically, the nucleus is not part of the cytoplasm. All right, so parts of cytoplasm include the cytosol, that's the intracellular fluid, that's the water inside of a cell. There's a lot of stuff dissolved in it. We also have organelles in the cytoplasm. These are little organs. These are structures inside the cell that help it do work, right? Each has a unique shape and function. And there are two types of organelles. They can be membrane-bound organelles or non-membrane-bound organelles. Membrane-bound organelles are, of course, surrounded by a membrane. And non-membrane-bound organelles do not have a membrane, such as ribosomes do not have a membrane. Membrane-bound organelles, yes, enclosed by a membrane, they separate the contents of the organelle from the cytosol. And this includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Non-membrane bound organelles are not enclosed within a membrane. They are typically made of protein or RNA in the case of ribosomes. And they include ribosomes, the cytoskeleton, the centrosome, and the proteasomes. Inclusions are things that the cytosol stores temporarily. They're not considered organelles. Mostly they're molecules that are added to or removed from continuously. And so examples include pigments such as melanin, glycogen, which is again the storage form of carbohydrates, and triglycerides. Here we have a model of a cell. Now this cell doesn't actually exist. There's no cell that has both cilia and a flagellum, right? So they're just showing you everything that a cell could have. And we do see a flagellum down at the bottom. There are also cilia, those hair-like projections, and then to the right of that are microvilli, finger-like projections that increase the surface area of the cell. In red, you see the mitochondria. The mitochondria are known as the powerhouse of the cell. They produce a lot of energy through the process called oxidative phosphorylation. And over on the left, big in blue, you see the nucleus. There's a double layered membrane there surrounding it. In the center, you find the nucleolus, which is responsible for making ribosomes. All right, and then of course inside of the nucleus you'll find DNA. Outside of the nucleus, you find the endoplasmic reticulum, and there are two types of endoplasmic reticulum here. Right up against the nucleus, you find rough rough endoplasmic reticulum, and it's called rough because it's got all these bumps on it, and those bumps are ribosomes. The smooth endoplasmic reticulum does not have bumps, does not have ribosomes. All right. Then another major one pictured in green up top is the Golgi apparatus. And the Golgi apparatus is responsible for modifying proteins and folding them into the proper shape before releasing them. It can also be used to store them. until the time is right. All right, plasma membrane. So it has a sort of fluid mixture composed of equal parts lipid and protein. They're equal by weight, but by number, the majority of it is lipid. And it regulates the movement of substances into and out of the cell. It contains several different types of lipids. Mostly it's phospholipids. There's also cholesterol in the membrane, and then glycolipids also. Phospholipids look like a balloon with two tails. We mentioned that before. They have a polar hydrophilic head, water-loving head, and two non-polar hydrophobic tails. All right. Sorry, my phone was just freaking out, getting a weird message there. All right. Um, Good. So the phospholipids, yes, they have the hydrophilic head and the hydrophobic tail. And they form two parallel sheets that lie tail to tail. Okay, so we call this the lipid bilayer because there are two layers of lipids in the membrane. All right. The hydrophobic tails form an internal environment inside of the membrane itself that is hydrophobic. The hydrophilic polar heads are pointed out toward the water. All right. So the phospholipid bilayer is the basic structure of the framework, and it ensures that the cytosol stays inside the cell and the interstitial fluid or tissue fluid remains outside. We also have cholesterol in the membrane, and it's scattered throughout the membrane. It helps strengthen it by preventing freezing at low temperatures and preventing the membrane from breaking apart at high temperatures. And then we have glycolipids, which are lipids that have carbohydrate groups attached to them, and they're located on the outer phospholipid region only. And it forms the glycocalyx or sugar coat that surrounds cells. And the sugar coat is read by immune cells. They come along, they feel the cell, and they say, okay, I recognize this pattern. That's one of my cells. I'm not going to attack it. Here we see the membrane, okay? And you can see the tails are oriented to the inside of the membrane, right? Okay, the reason for that is that they are hydrophobic, and so they are pushed out of the way by water, pushed toward this inner region here. The opposite end of these phospholipids is the hydrophilic head, which loves water, and it wants to be oriented toward water. So that makes this configuration really stable. If you add phospholipids to water and shake it up, you will see these structures forming spontaneously. All right. You can also see here the glycolipids, which are the lipids that have sugar groups bound, and these sugar groups form that glycocalyx, the sugar coat, on the outside of the cell that tells the immune system that this is a human cell or my cell. All right. So membrane proteins, they're about half the plasma membrane by weight. They float and move around in the fluid bilayer, and they perform most of the membrane's functions. There are two structural types, integral and peripheral. And if we go back up, you can see near the right side that big protein that passes all the way through. That is an integral protein. If you look to the left of it, there's another integral protein that forms a channel, and next to it is a peripheral protein. Integral proteins pass all the way through the membrane. Peripheral proteins may enter partially into the membrane, they may not, but basically they don't pass all the way through. That's the difference. So proteins carry out most of the specific functions. All right, so integral proteins are embedded inside the membrane, inside the phospholipid bilayer. And they have a hydrophobic region that interacts with the hydrophobic interior. In other words, they have a hydrophobic region that is perfectly comfortable being embedded in the hydrophobic region of the membrane. They also have a hydrophilic region, which is perfectly comfortable being exposed to the water, the aqueous environments on either side of the membrane. And many of them are going to be glycoproteins with a carbohydrate portion. Integral proteins are again not embedded in the lipid bilayer, and they are only loosely attached to either the external or the internal surface of the membrane. Transport proteins regulate movements of substances across the membrane. So examples here are channels, carriers, pumps, symporters, antiporters. Channels basically have an opening in the center that allows substances to pass through. Carriers change shape in order to move something across the membrane. Pumps require energy to force things against their concentration gradient. Symporters will transport two things in the same direction, either two things into the cell or two things out of the cell. And then antiporters pump things in opposite directions. One example of symport would be glucose. And so there is a glucose symporter that requires salt, right? So salt and glucose are both brought into the cell through the glucose symporter. They go together. And in fact, it's the salt gradient, the sodium gradient, that pushes glucose in. Antiporters push things in opposite directions. And so one example would be the sodium potassium ATPase, where sodium is pumped out of the cell at the same time that potassium is pumped in. All right, so we also have cell surface receptors. These are proteins on the surface of the cell that bind to ligands. What's a ligand? Well, it's anything that can bind to a receptor. The receptors have a specific shape, right? And so if the receptor has a shape for a square, only square-shaped molecules can fit in, right? Now, there's no receptor that actually fits a square. That's just a easy example. But the ligands are floating around in the fluid, and if one of them comes in contact with an appropriate receptor, it will stimulate that receptor and then get a response in the cell. So for example, neurotransmitters are released from a nerve cell, and they bind to receptors on a muscle cell. And when that happens, we begin contraction of the cell, contraction of the muscle. And then identity markers. These communicate to other cells that they belong to the body. These markers are used to distinguish healthy cells from cells that need to be destroyed. Sometimes when a cell is really sick, it will put markers on its surface that says to the immune system, basically, please destroy me. I'm too sick to go on. So functional categories of proteins still, we've got enzymes. Now enzymes are important. Enzymes do most of the work, most of the work in a cell. And they can either be attached to the membrane or they can be, to the internal or the external surface. There are also enzymes floating around in the cell. And they catalyze chemical reactions. In other words, they speed up chemical reactions. One way to think about enzymes is imagine you had a toy factory, right? And you wanted to put doll heads on doll bodies. Well, you could pay a worker to put doll heads and doll bodies in the same box and then shake that box for, 8 hours. And at the end of that 8 hours, you might have one or two dolls that got assembled properly. That's what it's like when we're waiting for reactants to float around and meet each other in the cell. An enzyme, on the other hand, is somebody who comes along and grabs a doll head and a doll body and then pops them together. The enzyme speeds up the chemical reactions. It facilitates the chemical reactions and makes them happen much more quickly. We also have anchoring sites on the membrane proteins. They secure the cytoskeleton to the plasma membrane. And then we have cell adhesion proteins, which attach cells to other cells. All right, so you can see all of these types here. I'm going to pause it on this video for just a second. All right. So moving on. We've mentioned the plasma membrane is a physical barrier between the cell fluid and the tissue fluid. Okay? Between the cell fluid and the interstitial fluid. And it regulates movement into and out of the cell. It establishes and maintains electrochemical gradients. I mentioned the sodium potassium ATPase earlier. That's the sodium potassium pump. It pumps sodium out of the cell and potassium in, but it pumps 3 sodiums out for every 2 potassiums it pumps in. And when it does that, it makes the outside of the cell more positive than the inside. All right. The membrane is also important in cell communication, communication with other cells. We communicate with the immune system. Hey, it's me, don't attack me, or hey, I'm sick, do attack me. Membrane transport is the process of obtaining and eliminating substances across the plasma membrane. So there are 2 categories of transport. They can be passive or active. All right. Now, for passive transport, this occurs without energy. Active transport requires energy. Okay, so passive transport includes diffusion and osmosis. Um, Basically, things are always going to diffuse from an area of high concentration to an area of low concentration. When it does this, we use the phrase diffuses along its concentration gradient. Okay. Now imagine you had a box of marbles, and all of the marbles were concentrated on the left side. They're all kind of stacked there on the left side. And then you start shaking that box of marbles, just kind of rotating it, rolling it around. Um, After a minute, you would expect to find that the marbles had moved and they were pretty evenly distributed throughout the box, right? Well, yeah, that's kind of how diffusion works. So these molecules are in there and they're bumping into each other, and so they're going to keep bumping into each other in an area of high concentration, and that's going to push molecules out into areas of low concentration until eventually we reach equilibrium where everything is evenly distributed. All right. And so in diffusion, there is simple and facilitated diffusion. Some things can get through the plasma membrane without help, and some things cannot. All right, so remember there's that hydrophobic zone in the center of the plasma membrane where the tails are. Water can't get in, other charged molecules can't get in, and then of course large molecules can't get in. So charged molecules, that would include things like ions. All right. So ions can't get through. What can get through? Well, small lipid soluble things, small neutrally charged things like alcohol or CO2 or oxygen, O2. Those can diffuse freely into the cell. They pass through the membrane no problem. And so that's simple diffusion. They just pass straight through the membrane. Facilitated diffusion requires a protein. Okay? Facilitated diffusion requires a protein to help the substance, molecule, whatever it is, get into the cell. And so the example here would be water. So water can't just enter the cell. It's polar. It can't pass through the plasma membrane. However, if a cell has channels, protein channels embedded in the membrane that let water through, water can get in. And in fact, we do have these protein channels. They're called aquaporins. and they allow water to enter the cell or exit. Good. So under facilitated diffusion, you can have channel-mediated or carrier-mediated facilitated diffusion. And with channel mediated facilitated diffusion, that's where we have a channel that's a protein with a hollow space in the center that lets a substance go through. So the example would be like ion channels. When they're open, they let ions flow through. There are sodium channels, there are potassium channels, there are chloride channels. Most of the time they're closed, but we can open them when we need to. And the ions will diffuse from areas of high concentration to areas of low concentration. So for instance, sodium, which is at high concentration outside the cell, that will diffuse quickly into the inside of the cell where it is at low concentration. Then you can also have carrier-mediated facilitated diffusion, and that's where a carrier protein has to change shape to allow a molecule to enter the cell. But the key here is with passive diffusion, we are always requiring that things move along their concentration gradient or down their concentration gradient. In other words, moving from areas of high concentration to areas of low concentration. One more example here, or one more topic here is osmosis. And osmosis is diffusion, but for water. And osmosis, or rather water, is always going to want to go wherever the solute is. Wherever the most solute is, that's where the water wants to go. And so if there's a high concentration of solute outside the cell, water will flow through aquaporins out of the cell. If the concentration of solutes is higher inside the cell, water will flow through the aquaporins into the cell. Now, what is... no, I guess that's good. All right. So that's osmosis in a nutshell. Passive processes. Now, active processes require energy, okay? And they're also going to require a protein, but they're going to require energy, okay? And we have active transport, which involves ions and other things being moved against their concentration gradient, typically through pumps, right? So you can have primary active transport and secondary active transport. Primary active transport is where we're spending ATP, that is using up energy, to pump something across the membrane. Secondary active transport is where we use a concentration gradient that we spent energy to create to our advantage. So what does that mean? Well, I mentioned the glucose symporter earlier. We can pump a lot of sodium out of the cell. We can use energy to do that. We create a concentration gradient where there's a lot of sodium outside the cell. And then we can use that concentration gradient to push glucose into the cell through a symporter. Okay, and that's secondary active transport because we're not spending energy to bring glucose in. We're spending energy to create the concentration gradient that will bring sodium and glucose back into the cell. All right. And then with secondary active transport, there is symport and antiport. And symport is where substances are moving in the same direction, and antiport where they are moving in opposite directions. One moves in while the other moves out. All right. One more active process. We've got vesicular transport, and a vesicle is a little sort of lipid bubble, right? It's like a really small lipid bubble. And we can perform exocytosis, where we have a vesicle that fuses with the membrane and dumps its contents outside the cell. One example of that would be like the release of neurotransmitters. We've got them stored up and sitting around in a vesicle. When it's time to release the neurotransmitters, that vesicle comes up, binds to the membrane, and then dumps its contents outside the cell. Endocytosis looks like the exact opposite process, where there's something big outside the cell we want to bring in, and then we form a vesicle around that thing and then bring the vesicle into the cell. All right, and then endocytosis can be further broken down into phagocytosis, which is literally means cell eating. White blood cells do this where they engulf a like a bacterium and then eat it. Penocytosis, where we're just forming a vesicle full of fluid. Penocytosis means cellular drinking. And then receptor-mediated endocytosis requires a receptor in order to begin endocytosis. That is, you have to stimulate a receptor to begin the endocytosis. All right. So we've talked about all these. The rate of diffusion, we should say, this is important to point out, depends on the steepness of the concentration gradient. That is, how big is the difference in concentration inside and outside of the cell. The greater the difference, we say it is more steep. More steep the concentration gradient is, that's going to increase the speed at which diffusion happens. Also temperature. So things are just moving around due to their thermal energy, right? The more thermal energy you have, the more molecules tend to move around, and so temperature will increase the rate of diffusion. Here we see diffusion where we have dropped some red food coloring into some water, and at first it is at high concentration in the very center of the beaker, and then over time it spreads out from the area of high concentration to areas of low concentration until over time on the right you can see we have reached equilibrium. The substance has fully diffused to all parts of the solution. All right. So we talked about all this. Tonicity, that's a good one. Tonicity is the ability of a solution to change the volume or pressure of a cell by osmosis. And there are three types of solutions. We say they are isotonic, hypotonic, or hypertonic. In an isotonic solution, you have the same concentration inside the cell and outside the cell. Iso means same. So normal saline, that's just salt water, has a concentration of 0.9% sodium chloride, and that's actually going to be about the same as your cells and your blood and your tissues. This is a commonly used IV solution. It is isotonic. A hypotonic solution has less solute in it than the cell does, okay? And in this case, where's the water going to go? It's going to go from the surrounding fluid into the cell, and then the cell is going to swell up. I had a teacher a long time ago say it would swell up like a hypo, right? And it may burst. Lysis is a rupturing of the cell, especially red blood cells. Hemolysis is the rupturing of red blood cells or erythrocytes. Then we have a hypertonic solution. Hyper means higher. Hypo means lower. Hyper means higher. And in this case, there is a higher concentration of solutes outside the cell, and water is going to leave the cell to enter the surrounding fluid where there's more solute. In this case, the cell will shrink, and this is known as crenation, the cellular shrinkage due to osmosis. You can see that here we have an isotonic solution. There's no net movement of water because there's the same concentration inside and outside. In a hypotonic solution, there is less solute surrounding the cell, so water moves into the cell. And in a hypertonic solution, there is more solute surrounding the cell, so water leaves the cell, and so it crenates or shrinks. All right, so we talked about the active processes. The sodium-potassium pump or sodium-potassium ATPase is an important one, and basically three sodiums bind to it on the inside. All right. And then a ATP molecule. These are the energy molecules that we use to do work in the cell. That's going to cause a change in shape of the protein, which pushes the three sodium molecules out. Then two potassiums come in, bind to it, and the protein goes back into its normal shape, which pushes the two potassiums into the cell. So the key takeaway from this is that three sodiums are being pushed out for every two potassiums brought in. And we're going to use that sodium concentration gradient for a lot of different things. All right, transport. All right. All right, so we talked about all of that. The plasma membrane establishes and maintains an electrochemical gradient known as the resting membrane potential. This is the difference in voltage across the membrane, the difference between the outside of the cell and the inside of the cell. And this is essential for muscle and nerve cell function. All right, so the electrical charge difference at the plasma membrane is the membrane potential, and the resting membrane potential is the difference between the inside and the outside when the cell is at rest. So there are two conditions for the resting membrane potential, and that's an unequal distribution of ions across the plasma membrane. In other words, more potassium in the cytoplasm, more sodium in the interstitial fluid, and this is due to the constant action of the sodium-potassium pumps. Also, the unequal relative amounts of positive and negative charges. So there are more positive charges on the outside of a cell, and there are a lot of negative charges in proteins on the inside of a cell. All right. And the resting membrane potential for each cell is going to differ, but for nerve cells, it should be about negative 70 millivolts there. All right. And this is kind of a complicated picture, but going through the proteins on the left, you have a potassium channel, which lets potassium out. It's probably, it's probably a potassium leak channel. To the right of that, you have the sodium potassium pump, which is pumping out sodium and pumping potassium back in. All right. Then you have another potassium leak channel, then a sodium leak channel, then another potassium leak channel. By the way, K is potassium, Na is sodium. All right. And so these channels work to get more sodium out of the cell and more potassium into the cell and keep that voltage at about negative 70 millivolts. Now, the sodium potassium pump is constantly running, and so we have the leak channels to try and keep the voltage steady. All right, so direct contact between the cells is important for some cells to function. So, for example, we have cells of the immune system which need to feel out the cells around them and distinguish between normal cells and either pathogenic cells or sick cells. Sperm and oocyte. The egg has a unique glycocalyx or sugar coat that allows for the recognition by sperm during fertilization. The sperm know to unite with the egg because of its special glycocalyx. And then cellular regrowth following injury. Damaged tissue can be replaced by cell division in the epidermis. And cellular contact prevents overgrowth. In other words, when the cells are touching each other, they know, like, oh, we should probably stop dividing. We filled in the gap. Most cell communication occurs through ligands, where the molecules that bind with macromolecules. Neurotransmitters from nerve cells and hormones from endocrine cells will bind with receptors. Those are ligands that bind to receptors. And they're important for controlling growth, reproduction, and other cellular processes. The three types of receptors that bind ligands are channel-linked receptors, enzymatic receptors, and G-protein coupled receptors. So here's a channel linked receptor, and basically when it detects the presence of the ligand, when the ligand binds, it's going to change shape and open the channel up. And so one example would be like the acetylcholine receptor. Um, in muscle, the acetylcholine receptor detects the neurotransmitter acetylcholine. Whenever you want to stimulate a skeletal muscle cell to contract, you're going to release acetylcholine. It's a neurotransmitter released by the nervous system, and it binds to acetylcholine receptors. And when they bind, it opens a channel up that lets sodium flood into the cell, which changes the membrane potential, which carries an electrical signal down the length of the membrane. But we'll talk about that in the muscle unit. All right, so there are protein kinase enzymes, and protein kinase enzymes phosphorylate other enzymes, which activates or deactivates them depending on the enzyme. And in this case, they're doing it when they are stimulated to do so by a ligand, by detecting their specific ligand. G protein coupled receptors are incredibly important, especially in pharmacology and understanding how drugs work. A lot of drugs, not all, but many drugs work by activating G protein coupled receptors. And basically, how this works is that the ligand binds the G protein and at least the G protein receptor, and that stimulates an intracellular G protein to go off and activate other proteins. For instance, it may go off and activate the protein adenylate cyclase, and adenylate cyclase will begin to produce a second messenger molecule called cyclic AMP, or CAMP, and that CAMP is going to go off and tell other proteins what to do. All right. And so in this case, the receptor doesn't actually the ligand doesn't enter the cell, but the ligand is the first messenger that binds to the G protein that causes a change on the other side where other proteins will be activated and will create the second messenger, CAMP, or cyclic AMP, which goes off to tell even more proteins what to do. Uh, CAMP starts what's called a cascade. All right, and it's called a cascade because one protein activates another, which activates another, which activates another. We don't need to get into all the detail, especially because in each cell or for each G protein coupled receptor, it's going to be different. All right. I am really kind of tired of talking. My throat hurts, so I'm going to take a break here and we'll come back to it later.